Hello, my name is Felix Schürmann from the École Polytechnique Fédérale de Lausanne in Switzerland. Together with my colleague Eilif Muller and our collaborator Professor Idan Segev from the Hebrew University of Jerusalem, I'm going to present to you the brain simulation platform. We will cover the science that went into building the platform, describe some of its functionality, and close with giving you an example of the novel science the platform enables and accelerates. Research in neuroscience, the data and understanding it produces, is highly fragmented. Integrating and unifying our knowledge and understanding of the human brain is a strategic goal of the Human Brain Project. It is a multidisciplinary challenge involving experimentalists, informaticians, simulation-based researchers, theoreticians, technologists, and cognitive neuroscientists. But how can we effectively integrate the contributions from these different disciplines? The Human Brain Project's solution is an end-to-end integration which propagates knowledge from experimental data to neuroinformatics to data-driven models, which are then extensively validated. These models form the reference for automated, quantitative, and repeatable simplification for neurorobotic and neuromorphic applications and hypothesis-driven studies of brain and cognitive function. To implement this vision, the Human Brain Project is developing 6 interconnected ICT platforms to integrate the activities from data upload and curation all the way to virtual neurorobotics experiments and make them accessible for collaborative research. The brain simulation platform provides a crucial link between experimental neuroscience and neuroinformatics on the one side and theories of brain and cognitive function on the other. This bridge is achieved by data-driven models which iteratively integrate multimodal and multi-resolution data in a hypothesis-agnostic manner and can be systematically validated and simplified for specific applications or to test specific theories. The platform relies on services from the neuroinformatics and HPC platforms, and provides models and simulation capabilities to platforms such as the neuromorphic and neurorobotics platforms. The technical challenge of the brain simulation platform is to allow integrative, iterative, and collaborative data-driven modeling, simplification, and validation, and therefore needs to address challenges of automation, reproducibility, and transparency. In order to prioritize and validate the technical choices during the ramp-up phase of the Human Brain Project, several scientific co-design drivers were chosen. They range from selected subcellular questions to specific brain regions such as the somatosensory cortex, the cerebellum, or the hippocampus, and also include the perspective of novel brain-wide datasets. In the ramp-up phase, those are focused on the rodent brain and human neurons. At the core of the brain simulation platform approach is the realization that our experimental data on the brain is inherently sparse. For the foreseeable future, experimental techniques will not exist to simultaneously measure all pertinent aspects of complex brain systems. Because of this, some signals we can measure, let's take for example the local field potential or EEG, we actually don't understand how exactly they come about. In this context, data-driven modeling can help. We can introduce principles from the data we can measure and use algorithms to consolidate constraints coming from multimodal datasets. The outcome of this process is models that describe the original data as closely as possible in a self-consistent integrated view that make predictions about gaps in the data and that link previously disparate datasets. Contrast to the original data, these models incorporate our knowledge in an active way, making it possible to test our understanding in silico. In essence, they allow us to solve the inverse problem, giving us causal insight into otherwise non-accessible processes. We were able to demonstrate this approach in an unparalleled team science effort published last year in Cell, and which thanks to the Human Brain Project has been made available in an online portal. On the example of the somatosensory cortex of rat, we implemented an advanced set of tools and workflows for the integration of detailed anatomical and physiological data of that part of the brain. Several fundamental scientific advancements were required, ranging from how to identify and compensate artifacts in the source data, how to resolve constraints stemming from multimodal data, predicting missing datasets, as well as algorithmic and technical advances for the simulation on supercomputers. The brain simulation platform is built on this experience and generalizes and extends these proven tools. It makes these tools available for other science teams to apply to their region of interest and to accelerate their research. In short, there are 3 main components to the brain simulation platform. First, many of the software packages that are being developed for the brain simulation platform and on which it is built are made available as open source software. Second, the brain simulation platform provides access to all of its tools and workflows in an online environment, fostering collaborative team science and transparent use of supercomputers. Lastly, the co-design drivers of the ramp-up phase present their results and workflows in dedicated collabs. On the one hand, this showcases the science they could accomplish, and on the other hand, they represent blueprints for new collaborative modeling efforts. I will now hand over to Eilif Muller, who will demonstrate in more detail functionality of the brain simulation platform on the example of the hippocampus.
Hi, I'm Eilif Muller, a researcher here at the Blue Brain Project in the EPFL, And I will demo for you now collaboratory assisted modeling of the hippocampus, a widely studied brain region known to be involved in cognitive functions such as learning, memory, and navigation. To begin, one defines the interdisciplinary team by inviting researchers to work together on the hippocampus model. The modeling process follows a workflow for data integration derived from that described in a recent publication by the Blue Brain project. Model reconstruction begins with high-quality neuronal reconstructions as shown here registered in the neuroinformatics platform. Neuron models are constructed using feature-based multi-objective optimization techniques as implemented by the open-source tool BluePyOpt, a software developed during the first 2 years of the Human Brain project. An IPython notebook makes this process interactive and transparent. Placing neurons into the hippocampus model is implemented in an interactive IPython notebook workflow. The workflow is thereby flexible and modular to support all brain structures and the integration of diverse neuroinformatics and literature sources. Building relies on the 25-micron Allen Mouse Brain Atlas provided as a service by the HBP Neuroinformatics Platform, which defines the coordinate system and regions of interest. Neurons are placed and oriented according to data on densities and composition from literature through a series of steps. After each step, the user has interactive feedback. Shown here, the 3 components of the neuronal orientation fields can be visualized and the morphological composition analyzed to quickly assess it conforms to the user's expectations. A framework for automated and repeatable validation analysis is also provided for systematic validation. To connect the network, a previously published algorithm is used, which is constrained by a variety of experimental data. Support to parameterize and launch the method on HPC resources is provided. State-of-the-art visualization web services provide a rich window to explore and refine the model. Once the model is constructed and validated, a simulation configuration and launch tool supports the user to run the model on HPC resources. Interactive analysis and visualization tools can then be used to investigate the results of the network simulation. A proof-of-concept automated point neuron simplification workflow provides a path to use the model with the neuromorphic and neurorobotics platforms. Further details on these platform capabilities and others can be found in the Platform Online section of the Brain Simulation platform, where focused documentation and screencasts help users get started. There is a vibrant scientific community focused on modeling in the hippocampus using diverse strategies. An active outreach program has established an ongoing exchange with leaders of this community so as to guide the development of the platform to catalyze and support integrative neuroscience for the hippocampus. Similar approaches have been applied for other brain regions, the cerebellum and the basal ganglia, and are available in their respective collabs. Thanks for watching this demo of team-based modeling of the hippocampus supported by the Human Brain Project Brain Simulation Platform. We welcome community collaboration to continue to develop and refine the model and to explore how it works. Please get in touch.
So I want to tell you about a very interesting story that emerged from this collaborative capability of the Human Brain Project of several groups, actually about 20 people from Amsterdam, from Madrid, from EPFL, and from Israel, from us, trying to, for the first time, understand human neurons. the functionality, the biophysical properties of human neurons. So here you see to the left a beautiful reconstructed human neuron. In the right, it's very fine synaptic location, the spines. And here is a set of reconstructed neurons from Amsterdam. All came from operations in hospitals in the Free University in Amsterdam. So we have a fresh data for the first time. And having this data, we were starting to use the collaboratory in order to build a reliable cell model, as I said, for the first time for human cells. So we have the physiology and we have the morphology and we have an automated protocol whereby we add specific electrical features, channel densities, type of channels into the model with an objective to generate a simulation of the cell activity based on its morphology and to optimize the response of the model as compared to the physiological response that was measured experimentally on these particular cells. So these are cells from the temporal cortex. You see the black spikes here. These are experimental spikes, and you are starting to running this automated multiple objective optimization algorithm that was developed under the Human Brain Project. And here you see the red spikes, which are theoretical spikes, superimposed beautifully, fitting very nicely the electrical activity of these real cells taken from the brain of patients. And here we do something that is very unique. We inject experimentally a small step current, as you see below, to a cell that you see on the left. You get a transient experimental response in black. This is the experimental response subthreshold for a very brief current. And then we are using the optimization techniques based on this particular morphology. And we found, to our surprise, never happened before to us in other cells we tried, that in order to fit this transient that you saw, you can only fit this transient theoretically, assuming that the nerve cell of human, this particular cell from the cortex of the human, can be fitted only if we assume that the capacitance, the specific capacitance per unit area of these cells is about half of rodents. We never got this, and this result is really true for the 6 cells that we have. And so this was a surprise. How come that we have different capacitance? So we went to experiments in Amsterdam and directly measured using what is called nucleated patch approach. And indeed, as you can see in the right, human cells have half of the capacitance compared to mouse cells. And this has an implications, functional implications, for the processing of signals in our big brain. Low CM, enhance signal transfer, enable better synaptic connections from the dendrite to the soma. What does it mean? It means that through this collaboration with Amsterdam, Madrid, Israel, and EPFL, we succeeded to find something new about human cells. And this was done through automatic modeling. This enabled us to discover something new, that human cells have a distinctive membrane properties. So I want to summarize in saying that brain research certainly requires a collaboration of teams. I could not do it myself. It would take me long time with old techniques. Now we can simulate cells very fast and very reliably and very beautifully, as you see, and learn something new. So these detailed simulations, which are essential step in my mind to understand a biological phenomena, go through the computer, building step by step the model you want to try to build, is an essential step towards understanding the brain. And one clear thing that we all learn, you have to do it together. We are going into a wonderful path. It's new, it's automated, it's shared by all. We collaborate as a team with one big mission, understanding the brain.